So I'm going to talk about reverse engineering the blood-brain barrier. So I should also say I'm an engineer, and the goal of one of our research projects is to build a blood-brain barrier on a chip. And I should say in advance as well that we haven't yet succeeded in doing this, and we have no new scientific breakthroughs to report, but hopefully you'll find something interesting in what we've done so far in getting to that point. Before I talk about some of our research, I'm just going to give a brief overview of the blood-brain barrier that may be useful. So, being an engineer, I'm interested in the structure and function of the blood-brain barrier. So first of all, one of the main functions of the blood-brain barrier is the vascular side in powering the brain. The brain requires about 20 watts to run, so about a dim light bulb. Interestingly to me as an engineer, the energy density of the brain is second only to the liver, so liver and brain are the most energy-hogging organs in the body, which to me at least is quite surprising since there's no moving parts. Now, in comparison to the heart, the lungs, the GI tract, which have a lot of moving parts, the brain has essentially no moving parts and yet is very energy dense. And in large part, that's because there's something like 100 billion neurons that need to be supplied with oxygen and glucose. There is effectively no storage of energy, oxygen and glucose in the brain. It's all on-demand power. And in the body, we have obviously the lungs are the transducing unit for processing oxygen, and the GI tract and liver are the processing units to generate glucose. And they're supplied by the heart, pumped through the vascular system, through the network of capillaries. So, in order to make sure that we're not limited in cognitive function by the supply of nutrients, but the rate of synapse operation, then each neuron has to be very, very close to a supply of oxygen and glucose. And to accomplish this, there's about 600 kilometers of capillaries in the brain. The capillaries are as small as 7 microns or so, and as many of you know, the endothelial cells in the capillaries wrap around upon themselves to form junctions with themselves and their upstream and downstream neighbors. The typical distance between a capillary and a neuron cell body is on the order of 10 to 20 microns, which means if you do a simple calculation based on the reasonable diffusion coefficient for glucose and oxygen, it means that the response time or the transit time is around a second or so in terms of supply. So basically the idea is you don't want to be diffusion limited in your fuel source to the brain. Okay, so that's powering the brain is fairly straightforward, but the brain is also a very sensitive organ, and it's obviously important to avoid fluctuations in water, salts, hormones, and so on. In the simplest case, fluctuations in salt concentrations can obviously interfere with the membrane potential and cell activation. Obviously, we need to avoid immune cell transport. The brain is in a fixed volume. It's in a box, the skull, and can't tolerate any significant changes in volume. So it has to avoid an inflammatory response. And for similar reasons, obviously, the transport of toxins and the pathogens have to be avoided as well. So we've evolved this system that, to me as an engineer, is very strange. That you want to power the brain so you have 600 kilometers of capillaries supplying oxygen and glucose, and yet at the same time you want to protect the brain from things in circulation, and yet you have this incredible length and incredible area of capillaries to protect. As an engineer, if I had to protect the brain, I'd want to make that capillary distance as small as possible, so the interface between the vascular system and the brain was as small as possible. So we've evolved this very complex system with a very specialized interface to be able to maintain brain security and power the brain. And I think a couple of quotes are relevant here. In particular, Dave Linden, who's a neuroscientist at Hopkins, says that the brain is a cobbled together mess. So he's referring, of course, to starting with the brainstem, and as we've gradually evolved, we've built more complexity to the brain. That's required more power requirements, and it's basically we're building extensions all the time. We haven't had the opportunity to start from scratch, or as Dave Linden likes to say, we're trying to build a performance car on a Model T Ford chassis. Okay, so a brief history of the blood-brain barrier. All the action was in Europe, mostly in Germany, around the late 1800s, early 1900s. Probably, as many of you know, Paul Ehrlich is most associated with the discovery of the blood-brain barrier. He injected dyes, trypan blue, into the vascular system of dogs and noticed that all the organs except for the brain were stained by the dye. And his conclusion was that, well, the dye just doesn't absorb, the brain just doesn't absorb the dye, which of course is not quite true. A little later, Max Lewandowski did a couple of experiments. He injected ferrocyanide into the central nervous system, observed it stained the brain, injected it systemically, and observed that it didn't stain the brain. So he begun to form a notion of the brain capillaries hindering transport of certain molecules. Then finally, one of Paul Ehrlich's students, Edwin Goldman, did the Goldman's second experiment, which is basically he repeated the Ehrlich experiment, injecting rats with trypan blue, saw that all the organs except for the brain were stained. Then he did the key reverse experiment of injecting trypan blue into the central nervous system and noticing that the brain was stained but not the other organs in the body. And this is sort of the origin of the modern notion of the endothelium of brain capillaries being highly specialized, of there being a barrier. Now, for those of you who are interested in the blood-brain barrier should know that the life expectancy of blood-brain barrier researchers is very short. The three originators of the blood-brain barrier met relatively early deaths, so I caution you if you're interested in research in this field. Okay, so the modern notion of the blood-brain barrier has evolved to the point where we consider the neurovascular unit. It's become recognized in particular over the last 30 years or so that regulation of the vascular system to supply nutrients to the brain and to regulate what gets in and out of the brain is done collectively by a number of cell types and is modulated by a number of other processes that I'll talk about a little later. So the key cell types, if we consider a classical model of an endothelial cell wrapping around upon itself, there are pericytes, obviously specialized smooth muscle cells that cover maybe 10–20% of the endothelium. The endothelium is completely covered by astrocyte end feet, which of course we know now modulate local supply of nutrients to the brain through signaling from neurons. So neurons that are working hard in some region of the brain signal their local capillary to say, hey, we're working hard here, we need more fuel, and triggers via astrocytes dilation of the capillaries to supply more oxygen and glucose. And here's an image from a mouse brain where you can see the astrocytes stained with GFAP typically here. Here's the vessel. It's about 3 or 4 microns in diameter in a mouse, and you can see this range of end feet coming from a number of different astrocytes surrounding the brain. Of course, paracellular transport is controlled by the tight junctions. So down in the lower left is a EM image of the ultrastructure of a rat brain, a cross-section through a capillary. There's one endothelial cell wrapping around upon itself, and you can see the tight junction there where the two membranes overlap each other, where they form the junction with itself. And this is a blow-up of a similar region, and the early work here was done in the late 1960s, early 1970s to visualize tight junctions. So this is a rat brain capillary. Here's the lumen here. Here's the brain parenchyma here, and the pericyte is illustrated. We can see the basement membrane around the endothelial cell is about 50 nanometers thick. And here is the junction, this length here, where the two membranes from the same cell are wrapping around to form the tight junction. And in this experiment, HRP was injected into the vascular system, and basically you can see where the tight junction is because the HRP has diffused into the cell-cell junction up to this point, but you can see there's no HRP diffusing further. So back in the late 60s, early 70s, this was the first physical evidence of a barrier between the vascular system and the brain. So I mentioned the tight junctions. Tight junctions shown here is a typical cartoon of a cell-cell junction. There's the adherence junctions and the tight junctions formed primarily by claudin-5 and other associated proteins. And the tight junctions, the claudin-5 has two extracellular domains that heterotypically and homotypically interact and form these beaded-like bands over some region of the membrane to prevent paracellular transport. Of course, transcellularly, there are a number of ways of getting across the membranes, either by passive diffusion or by some sort of carrier-mediated transport, receptor-mediated transport, absorption-mediated transport. This allows things that the brain needs to be pumped into the brain, but there's also a set of very specialized efflux pumps, with the canonical efflux pump being the PGP efflux pump, although there's many others in the ATP binding cassette family, that generally are polarized. So there's most of the efflux pumps, there's more on the luminal side than the abluminal side, and they basically recognize anything that can passively diffuse in that shouldn't be there is pumped out, and they're very effective. The array of efflux pumps are very effective in doing that. So now I'm going to talk from the other side of things. So if you want to get stuff into the brain, if you want to get a molecule or a drug into the brain, what are the strategies for doing it? And basically there's no good way. Since the discovery of the blood-brain barrier, there are very few drugs or molecules that do get into the brain that are not supposed to be there. Passive transport, of course, historically is the route that's been explored the most. And many of you may be familiar with the Lipinski rule of 5. There's many variations of that. But basically, molecules that have molecular weight less than about 500 Daltons and have the right balance of hydrophilicity and hydrophobicity described by water-octanol partition coefficient can get into the brain, although they may be substrates of the efflux pumps. Recently, Genentech and Hoffman-LaRoche have been exploring tricking the brain into using the transferrin receptor to deliver or transport antibodies across the brain. Recently, there's been a lot of interest in cell transport using monocyte stem cells as vehicles to deliver a cargo in the cells into the brain. This is all still at the research level. The Genentech and Hoffman-LaRoche work is at the clinical phase 1 clinical trial level. Another old field that's seeing a resurgence now is cell-penetrating peptides, of trying to exploit the ability of peptides to translocate, not to destabilize, but to translocate across membranes as a way of dragging cargoes into the brain. Back in the 80s and 90s, it was thought that inhibiting the PGP efflux pump would enable transport of molecules into the brain. As it turns out, that didn't work too well, in part because of side effects of some of these drugs, but also because of the broad spectrum of pumps, that you block one and there are many others, they have multiple substrates. So there's a move now to actually look for drugs that are inhibitors of multiple efflux pumps as a way of trying to improve brain penetration. And then there's always some crazy people over the years who are interested in transiently opening the brain by electric fields, AC fields. Ultrasound is currently in vogue, using emulsions of air bubbles and ultrasound to basically mechanically disrupt the blood-brain barrier. The rumors I've heard is that it gives one a massive headache. Okay, so let's look from a pharmaceutical point of view, just to illustrate some of these points for a minute. The worldwide pharmaceutical market for CNS diseases is around $80 billion. The top 5 selling drugs, at least in the US, are listed here. And what you notice immediately, of course, is they're all antidepressants or antipsychotics. And in fact, treatable CNS disorders are limited to mental disorders, plus chronic pain and epilepsy. And the reason for this is because these are all small molecules, and it's very difficult to get small molecules into the brain. There are no drug therapies for any of the neurodegenerative diseases, for neuroinflammation, neuroinfections. Brain cancer, for example, there's one FDA-approved drug in the US, temozolomide, and even that's not particularly effective. So whoever solves the problem of getting drugs into the brain, a 100-year-old standing problem, is going to make a lot of money. Just to highlight the point from passive diffusion, this is a plot of brain permeability from in situ brain perfusion experiments in rats versus the lipophilicity, the water octanol partition coefficient, just to illustrate that transport of many drugs into the brain, albeit in a rat brain, and there's obviously issues of comparison to humans, is roughly proportional to lipophilicity. There's a plateau here, and that's just because at very high lipophilicities, the concentration in the blood is not maintained constant. The rate at which it's sucked into the brain is faster than the rate at which it's supplied. The concentration decreases, and there's this apparent plateau here. But you can see 3 of the top 5 selling drugs are up here. Ethanol and caffeine, most people are familiar with the effect that they get into the brain rapidly. Glucose is a substrate for the GLUT1 transporter, so that has low lipophilicity but very high transport. Dopamine, morphine, and colchicine are substrates for the efflux pumps. Okay, from a disease point of view, many diseases of the CNS are associated with disruption of the blood-brain barrier, although there's not really a good definition of the blood-brain disruption, and I can't give you one. No one really knows what disruption means, other than permeability is increased, there's some kind of leakiness. But many diseases are associated with disruption, and there are obviously questions associated with, does the disease cause the disruption? Does the disruption then have a feedback effect on exacerbating the symptoms of the disease, and so on? And I've actually learnt a lot this week about the liver and hepatic encephalopathy and how that couples to the brain. Obviously, disruption of the blood-brain barrier can lead to bad things, changes in permeability, modulation of immune cell transport, and trafficking of pathogens. So in addition to delivering drugs to the brain, an issue that we're just starting to look at that I think is going to be important for the future is developing strategies and therapies to repair the blood-brain barrier. So how's one going to do this? To do this, one has to understand what the disruption is, its nature, and maybe develop stem cell-based therapies, for example, or find other ways for locally activating endothelial cells to proliferate, or enhance tight junction properties. Those are issues I'm not going to talk about today. I did prepare a brief slide after talking to Professor Hausinger earlier this week and reading some of his papers, but in the interest of time, I'm going to skip now to some of our work. So the reason we got interested in trying to reverse engineer the blood-brain barrier is the following. Over here on the left is the state of the art in measuring brain penetration, or trying to do drug discovery or determine whether a molecule will get into the brain or not. And it's a standard trans-well assay using MDCK cells. So MDCK cells are obviously canine, they're not human, they're epithelial, they're not endothelial, and they're kidney, and they're not brain. And in the 21st century, to me, as an engineer coming new to the field, that seemed pretty pathetic. So we basically started thinking about the neurovascular unit and what are some of the features, this isn't a complete list, and started to think about how could we build something that was more closely resembled the blood-brain barrier that might be useful both for fundamental scientific discovery and for translation to try to look at extravasation and intravasation, molecules or cells into or out of the brain. So some of the key features, of course, some go without saying: cylindrical geometry, shear flow, which plays an important role in modulating the blood-brain barrier, obviously other soluble factors in blood. The brain microvascular endothelial cells are highly specialized, as we've already said. as many of you may know, there are no good cell lines. Despite what the people who use the D3 cell lines say, there are no good cell lines for doing research, no human cell lines. Astrocytes are often used, or astrocyte extract, in this Transwell experiment, normally rat astrocytes, not human astrocytes. Astrocytes in rodents are very different morphologically and sometimes functionally compared to human astrocytes. Then obviously there's parasites and paracrine signaling and a whole load of other things. So this is a summary. I'm going to show you in a minute some videos of our starting point. As I said, we're nowhere near having a blood-brain barrier on a chip yet. We're a very small distance along the road, but I'm going to show you what we've done so far. So here is a cross-section of our platform. We basically have a hydrogel that represents the ECM, the extracellular matrix. We have a cylindrical hole down the center of it that we can perfuse media or blood or whatever we want through the vessel. We can introduce endothelial cells to seed on the internal surface, allow them to proliferate to form the endothelium. And basically we mount this on a microscope under physiological conditions. So here's just a brief picture. We use standard microfabrication technology, very low tech as it turns out, using PDMS, just a rubber plastic basically to define the channels. There's 3 in each one of the devices. Here's the ports. And within each channel there's one of these combinations here. And this is all, basically the perfusion is computer controlled. And here's the device on our microscope in our live cell chamber. So we started off doing some simple transport experiments. So this is a video of an artificial vessel on the microscope. It's lined with MDCK cells, so this is the starting point of just trying to see how we compare to conventional Transwell experiments. So you can see the vessel is, in this case, about a couple of 100 microns in diameter. You can see slightly darker here are the MDCK cells that line the vessel, so it's completely around this way. We're looking from underneath here. And in this experiment, we're introducing doxorubicin, which is a chemotherapeutic that's slightly fluorescent. So what you're looking here looping is we're flowing in, we're perfusing with doxorubicin, just as you would do in chemotherapy of administering the drug. And then what we're looking at here is both uptake in the MDCK cells and then transport out into the ECM. So we can do, for example, we can analyze the intensity out here. We can plot the intensity versus time, from which we can calculate the permeability. And we can measure the permeability in the wild-type MDCKs and MDCKs that are transfected with the efflux pumps and calculate an efflux ratio. So that's sort of a small step forward, very small step forward. So the next question is, can we do functional measurements using this platform? Can we have some readout that can tell us something either about disease or something fundamentally occurring in a vessel? So I'm going to show you now some relatively recent results looking related to cancer. So what we do here, and this is just a confocal image of a vessel where now we're using HUVECs, human umbilical vein endothelial cells. So human, we're getting closer to something relevant. And we're staining here for PCAM, a junctional protein, and DAPI in blue. And we're embedding cancer cells into the matrix. So we can embed either single fluorescently labeled cells or clusters of cells to mimic a small primary tumor. And we can flow. We use flow up to around 15 dynes per square centimeter, which is around getting up to the upper limit of flow in a typical capillary. And what we're looking at is a single fluorescently labeled breast cancer cell, so the MDA-MB231 cell line, which is very invasive. And what we're looking at here is the cell being attracted towards the vessel. This is classic amoeboid invasion of the local tissue. You can see it sending out these protuberances as it is digesting the matrix as it's going along. We're using relatively dense 4 milligrams per mil collagen. So this, we wouldn't expect to see contact guidance along large fibers. The matrix is too dense. So the cells are digesting the matrix, And this particular cell, you can see actually as it comes up to the matrix, it actually gets hung up. The two protuberances get stuck here around a large fiber that takes a while to digest. And you can see the protuberances here starting to sample the endothelium. So here's an example of intravasation. So obviously one of the first steps after invasion of metastasis, the spread of cancer. So what we're looking at here, this is just phase contrast. The slug-looking like thing is one of the MDA-MB231 breast cancer cells. Here is the vessel again, and you can see it migrating along the interface. It balls up. It exerts a force onto the endothelium that pushes the endothelial cells apart to open up a gap in the endothelium. It pushes through, and then the flow, which is going from left to right, just pulls it into the what would be the circulation. So here's an example of another phenomenon we can see here, the beginnings of angiogenesis. Here's a cluster of cancer cells adjacent to one of our vessels, and you can see that one of the endothelial cells here has become activated, is actually the beginnings of a tip cell. You can see two little tips, two protuberances being attracted up towards the cancer cells here, presumably in response to some of the classic angiocrine signaling factors, which was would be the first step of forming neovasculature in a tumor here. So those are just some examples. Again, no new science. All we're trying to do here is build a platform to see if we can recapitulate some of the phenomena that are known to be associated with metastasis. Of course, the details of many of the steps are not known other than from in vitro experiments or some intravital microscopy experiments in a mouse model. The point of us doing this is we have now human cells. Admittedly, there's many question marks associated with physiological relevance, but we can start visualizing some of these processes, and hopefully we'll be able to have some results that might have impact on the field later. So here's one way that we may be able to have more impact that is related to something more translational. So here again is an image of one of our vessels. This is a HUVEC-lined vessel. We're flowing in doxorubicin that I showed you again. Last time it was in MDCK cells. This time it's in human vascular endothelial cells. And you can see here in the phase contrast image is a single MDA-MB231 breast cancer cell down here. We're only looping for a couple of hours here. Before I was showing you 12 24-hour videos, so you'd see less activity of the endothelial cells. Here is the breast cancer cell. So we're flowing in doxorubicin just for 2 hours here, 100 micromolar doxorubicin. This is a relatively low concentration. It's about 2 orders of magnitude lower than a typical human dose. And the reason for that is the pharmacokinetics of doxorubicin show basically 2-compartment model with a very large distribution volume. So the volume, the concentration in circulation after half an hour or so is around 100 micromolar or so. So this is sort of a therapeutic dose. So you can see as a function of time, we introduce the doxorubicin. You can see uptake in the endothelial cells, and you can actually see uptake here in the nucleus of the breast cancer cell. And here's another video. This sort of shows you a little bit now how we can make a functional assay. So in looking at the efficacy of the drug, we look at proliferation. So here we've got, you can see a number of the breast cancer cells, and here is a blow-up is going to occur in a minute where we're actually seeing mitosis of one of these breast cancer cells. So we go and count mitotic events for 12 hours before introducing the chemotherapeutic, and we see 17% of the cells divide during that time period. After that, after treatment for 24 hours, we see no mitosis. So doxorubicin is an anthracycline, it's a DNA intercalator, so it's basically doing its job here. Okay, I'm obviously running out of time, so I just want to say a couple of words in terms of where are we going now? So you'll notice probably that there's still many, many challenges as we move towards something that might be similar to the blood-brain barrier. And I just want to show you a couple of things we're doing that we can start to incorporate into our platform. One of the main problems, as I said before, is cell lines. We've been using vascular endothelial cells that don't form tight junctions, but recently we've been able to reproduce a protocol that was reported by Eric Schuster's group in Wisconsin of differentiating iPSCs into human brain microvascular endothelial cells. Here we're using the BC1 cell line, iPSC cell line, which is widely used, and we've also repeated this in a GFP-labeled BC1 cell line. So we see all the tight junction proteins. There's 4 columns here. The BC1 iPSCs differentiated to BMECs. Retinoic acid upregulates tight junctions. GFP labeled version as well. So they all exhibit very good Claudin 5 expression, which no other cell line available for research in this area actually does. PGP pumps obviously highly expressed as well. If we look at the tier values, you can see that the differentiated BMECs with retinoic acid exhibit a physiological resistance of around 2,000 ohm centimeters squared. All other cell lines, MDCK cells, any other cell line that you can acquire has a tier value on the order of 50 to 200 if you're really lucky. So we now have a cell line to use that has physiological properties. The other issue, obviously, is starting to introduce other cell types, and we're particularly interested in astrocytes. Astrocytes in 2D look pretty deformed. In some cases, this is just on glass. You actually do see some protrusions, but they don't look particularly healthy, and this is pretty common. Most people use GFAP for identifying astrocytes, but as many of you know, quiescent astrocytes express relatively low levels of GFAP. It's only when they become activated that they overexpress high levels of GFAP. So we're particularly interested in introducing quiescent astrocytes into our platform. So we're not that interested in GFAP, or we're interested in trying to keep expression levels low. So going from 2D to 3D. So the brain, obviously very small amount of extracellular space. The predominant ECM protein is hyaluronic acid. There's no collagen in the brain. When we culture human astrocytes, fetal-derived astrocytes, into HA, 4 milligrams per milliliter, they're balled up. They don't look very physiological. And we're now using membrane stain because we're interested in trying to culture astrocytes that are quiescent, have low levels of GFAP expression. But after a year of going through seeding astrocytes in various gels, we stumbled across a gel that gives us very physiological morphology. These are 3D confocal images, and if we look at the GFAP expression version here, so this is activated, you probably couldn't tell that from an astrocyte from a human tissue section. The size is around 200 microns, about the same as human astrocytes. And you can see that in the right gel, so inactive, active, left and right, cell membrane stain here, and GFAP expression here. So with the right ECM, we can get physiological morphology and low levels of GFAP expression. With the wrong gels, we can get the right morphology, but highly activated astrocytes. So this is, in conclusion then, where are we? And I would give us probably a C- or a D in how good we're doing at the moment. So cylindrical geometry, yes, we've got it. Diameter, we're still at a sort of post-capillary venule type of size, about 100 microns. We need to be down, obviously, around 8 microns. We have no idea how we're going to do this at the moment. We've been thinking about this for many years. We don't have a good solution. Shear flow we can incorporate into our model up to physiological values. We are not using blood, but it's pretty easy. We've done this. You can add dextran to get the viscosity right, or you can use blood if you want to. Soluble factors we can add. I've shown you experiments with drugs. Human brain microvascular endothelial cells, we're nearly there. We've got the cell line. We need to figure out how to seed our vessels. 3D ECM, possibly. We're going to switch to the matrix for astrocytes that we've developed. Astrocytes, we're nearly there. We've got the right morphology, and we've got quiescent astrocytes in culture, not in our model. Pericytes, no. Paracrine signaling, no. Neurons, no. So we're a small way along the way, and I'd like to finally acknowledge my main collaborators, Alfredo Quinones and Dwight Burgles in neuroscience, and thank you for your attention.